So I like to start with this picture as it shows our ability to balance, even maintain straight gaze in a moving vehicle. And this is because we all have these three tubes in our inner ear, in our vestibular system. These are the semicircular canals. The salty fluid shown here in blue, called the endolymph, moves through the tubes when our head rotates. And this rotation results in bending of this gelatinous structure called cupula, which in turn results in bending of the hair cells that sit at the base of the cupula, which turn this mechanical motion into neural signals for us to be able to maintain balance. And this fluid flow in these tubes can be described with Poiseuille's law. So you can see that the shape here is extremely important. The flow depends to the fourth part, to the canal width, and inversely proportional to the length of the canal. So even small differences in this width or the shape of the canal has huge consequences on canal function. So if the canal is too long and narrow, then the drag of the fluid is high, causing it to be less sensitive, damping the motion. If the canal is too wide, then the fluid runs more freely, causing it to become overly sensitive. So there is a balance between the length and the width of this tube and the viscosity of the endolymph that's flowing in these canals. And it's for this reason this organ doesn't scale allometrically as most other organs do. So this is a graph showing that while the mass of the mammals can vary 7 orders of magnitude about, the size of the canal varies roughly only tenfold, because nature is trying to optimize the size of these canals for the sensitivity of rotation. So all this
That's
is
a question
about
the diameter and
so this is reflective in the length.
I understand that's
R,
right?
Yeah.
The diameter itself, does the diameter remain constant when you go
This?
from one or yes?
Oh, in the scale. In the scaling, yeah. It's not. I don't know if it's tighter or more spread out than this R. Yeah, I'm not aware, but that number is known. I just am not sure. Yeah, but it definitely doesn't spread like so. It's much tighter than that.
Genetically, when circular canal?
Yeah, so I'll come to that. Yeah, I'll come to that. I won't be able to talk much about the Evo part. I'll do my best to talk about the Devo part, but there's one slide. Yeah. Okay. So all this is to say that the shape of an organ is really important for its function, and we think canals really epitomize this form-function relationship. So how do these structures form? So this is the Devo part. And this is my one slide for the evolution. So jawless vertebrates, lamprey, hagfish. So structures without jaws look scary. They do have canals. They have 1 or 2 semicircular canals. Jawed vertebrates like us and like fish, frog, chicken, and mice have 3 mutually orthogonal canals. And this architecture of roughly mutual orthogonality is quite conserved across species.
Benthic organisms which are not agmathic also have 2 canals. And so where do they fit?
I'm not sure. Yeah, there are. So beyond these canals, that is helping us to sense this angular acceleration, we also have otoliths, and that several invertebrates have otoliths. So squids would have otoliths. That is a much more, I would say, ancient structure than semicircular canals. Yeah. It is an evolving structure. Like a fun fact is the angle variability that you see between the canals is becoming larger and larger in tree hanging sloths because they don't move very much. So they don't have a constraint to make these shapes very accurate, the way we do, or the way other animals that displace or move a lot do. Okay, our favorite model organism to study them is zebrafish, because zebrafish lack middle and outer ears, and their embryos are transparent. So it makes the structure that will give rise to the semicircular canals both optically and physically accessible. So you can already see some structures here. This is the otic epithelium, which is the embryonic inner ear. So this is the beginning of the life of the inner ear. It starts off as a thickening derived from the ectoderm. This thickening is next to the hindbrain. And this placode undergoes cavitation due to influx of water and ions that results in building up of hydrostatic pressure. And so this movie was collected by a former colleague of mine in my postdoc lab, in Sean Megason's lab, and Maha contributed theory to understand how this pressure can drive expansion of this otic placode. And this is to say that while the lumen volume is increasing significantly, the tissue volume here is not increasing proportionately. So really the fluid that's being accumulated is driving expansion through little contribution from the overall growth of the tissue. So this continues to happen from about when the fish is 15 hours old till about 2 days old. And it results in the formation of this tight epithelial structure. It's a single lead structure, yeah? The junctions between the cells? We don't know. Good question. We don't know how the fluid. It's likely both. Kishore, the first author on this study, he was collecting some time lapses to look at where the fluid is coming from, and he could see with a dye that it can go through between the junctions, but we also have evidence to say that aquaporins and it can go through aquaporins through the epithelial layer too. Through the cells? Through the cells, yeah. Yeah. Do you expect the volume of the cells to change as this process happens? Again, good question. It's not changing though. Yeah, it's not changing. But it's a passive transport, and there are for epithelial layers that are acting as barriers, there are several reports that show that you can still have water transport without change in cell volume. And from this single layered structure, this tissue will undergo a dramatic remodeling to give rise to the semicircular canals. So let's observe what happens in this transition. So instead of a 2D section, I'm showing you a 3D rendered view. It's an ellipsoidal shaped structure, and we're looking at the top half of this ellipsoid from the inside. So you can already morphologically distinguish where this embryonic structure is, and it has this invisible lumen. It is very similar to the endolymph constituents that will be there in the adult canal structure. It is already at this stage a salty potassium-rich fluid inside. Okay, so morphogenesis of these canals begins when different regions in the epithelium begin to bud into the lumen. So this is the apical surface of the cells that's budding in. The basal surface is facing the outside. And I'll let this movie loop so you can appreciate again what's happening. There is a topological remodeling, or 3 topological transitions happening, where you start off with a single continuous sheet, and then this bud pair, this anterior bud pair, will make contact. Posterior bud pair will make contact, and out of the plane, there is a ventral bud pair that will make contact.
When these tubes or when these buds form, are they already luminized, or do they luminize after they fuse?
What do you mean by?
Are they hollow?
They're not hollow, and I'll come to that. Yeah, they're not hollow. And they form these buds. Once they make contact, they fuse. It's not membrane fusion, it's formation of new cell-cell contact. They form these 3 pillars. These are not the canals. they are the hubs around which the canal is being demarcated. So this illustration helps to imagine it a bit better than those images. So instead of 3 bud pairs, I'm showing a single bud pair. The buds extend, fuse to form this pillar-shaped structure, and it's this tube around them that becomes the semicircular canal.
So, so the extension, the bud extension and the pillar that in the cartoon, which shows a sort of just a shell, but that is actually filled with cells on the inside?
It's not a cellular fill. So that's, I'm going to summarize quickly the work that I did as a postdoc and one of the first studies we did in the lab, which is to ask, how is this process initiated? What's inside those buds and then the pillars? And I can summarize to you that the classic modes of morphogenesis that we're used to thinking about, which is either local or global cell proliferation or some directed movements, rearrangements of cells, these are not happening. In fact, there's very little growth happening in this tissue, similar to the early stages where most of the expansion can be explained by just hydrostatic pressure. The morphogenesis of these buds is locally initiated by a component of the extracellular matrix called hyaluronic acid. It's a non-protein component. It's all sugar. It's the cells using these enzymes can make large chains of this polysaccharide. And one thing which you may already know, this is heavily charged. It has these carboxyl groups that attract sodium ions. It attracts counter ions, building a polyelectrolyte that swells with water significantly.
You can have divalent polyelectrolytes, which are calcium and magnesium cross-linked, or sodium, which will not.
It's usually sodium. It's usually sodium. Yeah. So there's no cross-linking. Yeah. Exactly. So it is really a charged, osmotically active gel. And I'll come to that, an experiment we did recently to condense these polymers in vivo, something that I couldn't do as a postdoc, but we were able to do now. But
evidence that divalent cations play any role, like the calcium?
Free-floating calcium outside, it can be there, but it's not dominant as sodium ions that are free in this extracellular space.
And sodium and not potassium.
It's usually sodium and not potassium. Potassium is rich in the endolymphoid. This is the perilymph side. Yeah, exactly. So yeah, the sodium-potassium transporters, the directed transport of potassium is towards the lumen, and this is the hyaluronic acid is in the extracellular space, which will become the perilymph later. Yeah. So just quickly for the developmental biologists or biologists in the audience, the genes required for hyaluronic acid synthesis are exclusively expressed at the sites of morphogenesis. So this is required for substrate synthesis, and this is required for the polymerization of the substrate. And I didn't mention in the previous slide that there are proteoglycans that can bind hyaluronic acid, and it confers the hydrogel some structural properties. The chondroitin sulfate side chains of these proteoglycans can also contribute to the osmotic swelling of this hydrogel. And we knew from literature too that the genes required for one such proteoglycan, versican, are also exclusively expressed at these sites that are budding from the epithelium. And we can knock these out and find that this is completely sufficient to block morphogenesis. So you don't get any budding morphogenesis if you knock out the gene required for hyaluronic acid substrate synthesis or the protein part of the proteoglycan.
The relative location of the 3 buds is already approximately orthonormal when you start out, or does that get?
So the location of the buds is very stereotyped, very reproducible where they start. There is a spatial information that on an ellipsoid, they're mostly on the dorsal part where the buds originate from, and there is a temporal information in the sense that the lateral buds form first, then anterior, then posterior, and the ventral ones there's a ventral bud that starts much later, but the information for for where it's supposed to be is already there in the form of a developmental factor, which
What is that?
what factor or what information?
Oh.
Yeah, I'll get to that. We don't know much about patterning though, so it's going to be very underwhelming. Just as a warning. Yeah.
In the embryo at this stage when the canals are forming, are they already are they able to swim? What's the environment?
Yeah, so these they're still called embryos at 2 days. They hatch naturally between 2 to 3 days. So they are able to swim. The earliest we can measure a behavioral response, like for example, for if there is a canal defect, is actually between 3 to 5 days. It's not reliable, but it's there. I can direct you to the tutorial I gave last week where you'll see that all the body axis, the tissues that will give rise to future organs are already in place. So there is a heart, there are eyes, everything is there. So they are just specializing in these forming these functional structures. So at the same time, heart is undergoing exquisite morphogenesis to form trabeculae. The eye is undergoing exquisite morphogenesis to form the optic cup. But all the germ layers and the primordial structures that will give rise to those tissues are already where they're supposed to be at this stage.
Also, they help stabilize orientation.
Every,
They do.
balance, like,
Still.
yeah, is there, yeah.
So if you have, if the embryo is experiencing flows in different direction while these are forming, the canals still form, okay?
Good question. We don't know the answer to that. Yeah, I'm happy to talk about that, like role of shear forces. I can try to get through very quickly in the first 10 minutes, like just so you have an understanding of the kinds of questions we ask in the lab, and then we can get to the fun stuff on what can we think about as these are developing, what kind of external forces could come into play, or environmental cues can come into play. So just to get through this, coming back from behavior to these to this charged polymer. So we're going back to the molecule here. So this is just to show that hyaluronic acid, and this is the chondroitin sulfate side chains. They are just underneath the cells that are budding. So these form very viscous gels, such that if we inject different sizes of dextran, only the smallest dextran percolates in this bud ECM. So there are no cells there. It's all this viscous goo of sugars that's there, and possibly some growth factors and morphogens that are trapped there. So if you remove Versican, all of hyaluronic acid is gone. So Versican is really needed to retain hyaluronic acid in the bud ECM. And what hyaluronic acid itself is doing, I already mentioned that it forms this charged polyelectrolyte that can swell. If we digest hyaluronic acid, this bud ECM collapses and buds fail to extend. And we concluded here with this and several other experiments that indeed hyaluronic acid is driving morphogenesis of these buds through osmotic pressure. But one criticism or one question I would always get is, but hyaluronic acid is not there, so you don't know it might be playing some chemical signaling role. So a postdoc in my lab did this experiment that was my dream experiment, where he injected this charged polypeptide, protamine. It has several arginine groups on it, and that precipitates out hyaluronic acid. So it's still there. The hyaluronic acid is still there, as you see in magenta, but it's no more behaving as that polyelectrolyte that can swell with water. So here you see the bud kind of collapse. They don't have a swollen bud ECM anymore, and the buds fail to extend. Okay, so this is happening through synthesis of hyaluronic acid when certain cells decide to start expressing these enzymes. But how is that pattern defined, which is your question, Maha? So we knew about this transcription factor from the literature. It's called LMX1B. It's a limb homeobox containing transcription factor. It's very well known in limb development. And it was known for its role in upregulating, promoting ECM synthesis, including hyaluronic acid and proteoglycan synthesis. And again, we knew from literature and we validated it is the genes, there are the paralogs, LMX1BB and BA, they're expressed at the sites of morphogenesis. And when we knock this transcription factor out or knock it down, knock down both together, then the expression levels of both hyaluronic acid and versican synthesis genes go to almost background level. But that's all we know about the pattern so far. We don't know why it's set up in this. So the LMX1BB, which is the more important one, is expressed in the entire dorsal region, but the buds are local, so we don't know.
When buds form, you always get three? Question, and do you get them simultaneously, or can you?
There are three bud pairs happening in the spatiotemporal order that I mentioned.
You never get more or less?
No. Unless it's a mutant.
Okay.
Yeah.
But it's not like a few start and then.
No, it's very reproducible. Very, it's like, we've never seen an embryo with more than 3 canals or less than 3 canals unless it's a mutant.
Maybe
or buds, yeah.
But buds.
No. Yeah.
Size of the original sort of tissue, if it's the other mutants, in the mutants where you do see different sizes or number of buds, the original tissue smaller or bigger?
It's complex. It depends on the mutant. Yeah, we have a mutant where you can anteriorize the aortic vesicle, and then the posterior canal becomes anterior-like, and the overall shape of the ellipsoid, of course, it's not an exact ellipsoid. It has all its 3 axes, anterior, posterior, dorsoventral, medial, lateral. Those become anteriorized. And there is one mutant in which actually, I don't know in the one with absent canals what happens to the overall size. We should look, yeah. These are reported mutants. We haven't systematically gone and looked at the development of these yet, yeah. Okay, just to summarize that, beyond cell behaviors, this structure is a good example of how extracellular matrix remodeling is driving morphogenesis. So here I'm going to talk about a recent study that we pre-printed to show how these buds are extending. And all this work was done by a really talented postdoc, Yusuke Mori, the first postdoc in my lab. So one thing I didn't mention is that as this osmotic swelling of the ECM is happening, the overlying cells are stretching significantly. As I mentioned, they are not dividing, but they are stretching. And this is in response to hyaluronic acid, such that when I digested this ECM, the cells lose their stretch. So we wondered, can this stretch be mechanically transduced? And a candidate, sort of an obvious candidate we had in mind was this transcription co-activator YAP that can respond to mechanical cues, including ECM stiffness, compressive or expansive forces. And our neighboring friend lab, Michelle Banya, generously shared a knock-in reporter line with us. And indeed, we see, one thing I didn't mention, YAP, when activated by these mechanical cues, gets dephosphorylated and goes to the nucleus. So nuclear YAP is reflective that it's active. So indeed, in these budding cells, if you see here in pink, we see lots of YAP. And this is dependent on presence of hyaluronic acid, such that when we digest, when Yusuke digests the bud ECM, you see the bud collapses and you don't see a lot of magenta here. So the nucleocytoplasmic ratio of YAP goes close to 1 in this condition. So what is YAP doing in this context? YAP usually functions by activating its target. It itself is not a transcription factor, but when it goes to the nucleus, it binds to transcription factors like TEAT that can then express several genes that affect cell behavior, like cell growth, changing fates, and so on. So how is YAP functioning in this system? We had created, during my postdoc, a single-cell RNA-seq atlas of the developing inner ear. And since then, including the work done in Sean and Alon's lab and other labs, there have been several publicly available embryo-wide atlases that we had access to ask what could be the potential targets of YAP. And in this, unanimously, among the top 5 genes was this gene, CCN1L1. It's a matricellular protein that is a known downstream target of YAP. In fact, this picture is from a review, a very old review, and they listed it as one of their key targets. So, and the expression of this YAP target is where we expect it to be, where morphogenesis is happening, where these buds are forming. So is indeed this CCN1L1 a YAP target within the developing semicircular canals? So yes, indeed, if we remove this node, if we remove osmotic swelling, in controls, you see that the expression of CCN1L1 increases, but when we digest ECM, this expression fails to increase. If we downregulate YAP, the expression of CCN1L1 is also reduced significantly. So what is this pathway doing here? I told you that there is almost no cell proliferation happening, so much so that if we block cell proliferation for 12 hours, morphogenesis still continues. And one of the most known functions of YAP is cell proliferation. So what is YAP doing here? We found that downregulating YAP or CCN1L1 abrogates morphogenesis. So the buds are much smaller, and they never manage to form these pillars that are ultimately demarcating the canals. And this is happening through the expression of hyaluronic acid and versican synthesis gene, such that when we downregulate CCN1L1, their expression level goes to background. And this function of YAP promoting ECM is actually a more ancient function of YAP. There is this beautiful paper from Thibaut Brunet's lab on choanoflagellates, where they show that the rosettes of these choanoflagellates are much bigger when they hyperactivate YAP, and this is through increased ECM synthesis. So what we think we found is this positive feedback loop, where after this initial synthesis of hyaluronic acid from this transcription factor, there is activation of YAP, which results in CCN1L1 activation that then results in more hyaluronic acid synthesis. This is nice, but why do you need a positive feedback? What is this positive feedback doing? So we wanted to explore the dynamics a little bit better of why do you need a positive feedback. Maybe, I mean, when I started, one can also just hypothesize that this is enough to explain why buds, how buds extend, that you just have this initial signal and then the buds grow. So we tested these tenets of what a positive feedback should look like. And one thing I don't have time to show, but we can ablate any node of this pathway and it affects all the other components. So we can knock down this and it affects this and so on. Okay, so is this positive feedback amplifying some initial signal? So one thing I didn't emphasize is that when buds grow, they are growing by recruiting cells from the side. So they're pulling onto the cells, and that's what's leading to increase in cell number. And this is dependent on swelling of hyaluronic acid, such that when we digest the matrix, there is no increase in the cell number in the bud. The bud just stays as it is initially. And we wanted to keep exploring this dynamics of what this recruitment looks like, how it's going, how these cells are being pulled into the buds. And we collaborated with our now longtime collaborator, Edwa, and a postdoc in his lab, Paul, who built a vortex model of a transversal section of this epithelial bud. And we knew these parameters from my postdoc work, the apical, basal, and lateral tension in these cells. And these cells are experiencing pressure, quote unquote, from the expanding ECM area that they're enclosing. And Paul regionalized this section where there are tip cells, budding cells, and these border cells that as the ECM is expanding, these cells are the ones that are getting recruited. And what he found was that as the cells are getting recruited, they significantly stretch at the bud border. So he he observed these transient overshoots in the aspect ratio of just these border cells, but not the tip cells. So here are the tip cells. They don't have these aspect ratio overshoots. We wanted to test this experimentally. Are we seeing some regional patterning of the cells here in the form of stretching? So Yusuke collected these beautiful time lapses with the YAP reporter line. So here you can just observe the nucleus first. You can see the nucleus stretches out significantly as it gets recruited into the bud. And now if you look at YAP activity here, it is highest when these cells are turning the corner. So this was really nice. Indeed, there is a mechanical patterning of cells happening as they're turning the corner. So he tracked several cells and found that the maximum stretch they experience and the maximum YAP activation they experience is happening at the bud border. And then this cell, once it turns, it becomes a budding cell, and eventually YAP activation decreases just by natural decay.
How does this translate in the time that it takes to make the stuff that they need to?
Yeah, it actually works beautifully. It's a really good question. So the time it takes to make a bud is roughly 8 hours. And the first cells that are specified are really 4 to 5 that have LMX1B. So when Yusuke was trying to see if CCN1L1 expression is affected by osmotic swelling, he had to digest the matrix twice to see a transcriptional change, because for YAP to go to the nucleus, then activate its transcriptional target, and for that to ECM synthesis, you can imagine 2 to 3 hours. So it has enough time to recruit 2 rows of cell, and that's basically it. You need like 4 total rows of this bud.
So you think they are producing this material in the end after they've made it through the corner or at the corner?
I think they start already. So actually, this was something I made an observation as a postdoc, and I didn't know what to do with it. The expression of Has3 is lowest at the bud border. So this is reflected, and CCN1. So this is reflected that they're just beginning to express CCN1. Yeah, yeah.
So I had 2 questions. One is the bud as it grows doesn't balloon out as a sphere, but rather as a tube. So that suggests there may be some kind of tension.
Yeah, I glossed over it. This is the work that I did as opposed to, I didn't even gloss over. I didn't mention it at all. We see these tethers produced by the cells. They are biased in circumferential axes, so they're containing. Yeah. So yeah,
The thing is, your cartoon showed that there was basement membrane, so presumably there is integrin adhesion and de-adhesion program.
Absolutely, yeah. So again, I didn't go over it just to save time, but indeed, when I showed where hyaluronic acid and chondroitin sulfate is, I didn't point out, but laminin is very low inside the bud ECM, but on the side there is laminin. So these cells here are attached to the ECM, and that's actually key for why we think the cells stretch here, because they are stuck to the basement and being pulled in. Yeah. Yeah. So yeah, we are exploring exactly this, whether there could be some attachment here. So we think that this feedback loop is amplifying this initial signal by recruiting more cells into the bud through this relay of YAP activation, which is producing then more ECM. And I'm going to try to speed through the next part, so we have time for discussion for more ongoing stuff. We wanted to ask if it can.
Then this, can you go back to this side?
Yeah.
What you call, I mean, it looks like you have a treadmilling of cells entering the canal. That depends on the referential you use. Would likewise say that actually it's the basement membrane that gets degraded at the base, and that actually.
I'm sure you're not the reviewer, because that would be a conflict, but we are asked this question on how the cells are relatively moving from on the side. Are they sliding? Are they degrading? What are they doing to the basement membrane? So we have from Ken Poss's lab a laminin reporter line, and we are doing FRAP experiments to see what happens to the basement membrane.
You think the whole cell
We don't see any.
shear between the basement membrane?
Yeah,
and the whole
yeah. I think they kind of like attach, detach, but I don't imagine it to be some kind of active migration. We don't see any part of the cell that has like a front or a rear. Yeah, so I think it's just passive, but there is something special here. There is integrin-based attachment most likely. Yeah.
I think either you degrade and it's local, or you have a complete shear and it's
Yeah, it is. It's similar to Claudio and Anais' like the adhesion deadhesion at the. Yeah, it is similar to that model. Okay, so is it accelerating the rate of bud extension?
Extension. Is that continued to the osmotic swelling?
Yes, it's the hyaluronic acid dependent swelling of the ECM.
More help. More hyaluronic.
Exactly. They increase ECM volume. And if we break the feedback, this is in the model, and this is with experiments. If we break the feedback, the rate at which this increase in ECM volume is lower. Yeah. Yeah, and this is true for bud lengths too. So one can ask, I mean, this is a philosophical question, and I'm just going to take a slide to speculate that why do you need a certain rate of bud extension? Like the process can take its time. The buds can meet. Why do you need a positive feedback to accelerate the system? And we know that positive feedback have a lot of features. They can buffer if there are varying initial concentrations. Through this feedback, you can buffer varying initial concentrations, and I'll come to that experiment. But just a short detour that I want to take, that why we think the rate of bud extension is extremely important is I want to remind you of the slide that I showed that initially the aortic vesicle is expanding because of pressure. We didn't know whether this expansion continues during the course of canal morphogenesis. And using unit pipeline, a student in the lab, Jia Chang, analyzed several aortic vesicles over time and found that indeed he could automate the lumen segmentation, and he could find that over time, this is still increasing. The lumen is still, the aortic vesicle is still expanding. And I haven't shown the data here, but we know that there's very little growth happening. So we think that the hydrostatic pressure within the aortic vesicle either stays there or even increases during the course of canal morphogenesis. So these buds, in essence, have to counteract the force from the pressure of the lumen. So if they're too slow, they're not going to meet each other. And this is something that we tested.
They meet in an expanding universe.
They meet in an expanding universe, exactly. Yeah, very poetic. Yeah, so Yusuke did this dose-dependent, let me keep track of time, I'm already out of time. So Yusuke did this dose-dependent reduction of CCN1L1 and found that only at the strongest dose would you abrogate morphogenesis, but in these intermediate doses, morphogenesis still continues. So you just need a little bit amount in the beginning to kickstart the process, and it's enough to let the process keep going. Another feature that we know that positive feedbacks do is that they can destabilize. So how is this being shut down? We had hints from literature that the process is shut down when the buds meet each other. So it's very critical that they find each other. And this meeting activates a GPCR that then shuts down hyaluronic acid synthesis through cyclic AMP signaling. And this was a really good test of the positive feedback model. So when we use this mutant, Yusuke finds that the buds overgrow significantly. Their volume increases nonlinearly. The CCN1L1 expression and Hastrian versicant expression is all there. So the buds keep growing. So indeed, the termination signal is coming from when the buds meet each other. And I'll show you an experiment that shows that this is sufficient to terminate the process, just the buds finding each other. So I'm not going to go over the summary because I feel like you got the gist that you have this feedback loop driving extension, and you have this signaling pathway that gets activated that terminates this process when the buds meet each other. So I just want to describe two sets of experiments that will summarize 2 ongoing projects in the lab that I'm very excited about, and we hope that we can write them, get feedback, and so on. So one thing I want to point out, there's lots of variability in the system, and we heard amazing things about variability from Adrian this morning. And I'll quickly point out one kind of variability here. The zones at which buds fuse to one another vary significantly. So they can meet tip to tip. This bud overgrew a little bit. It's going to meet its partner bud side to side. In this auric vesicle, we've ablated the auric vesicle. This bud is going to meet its partner bud earlier because the auric vesicle is smaller now and on its side. And we can do statistics on this, on where they're meeting, and it is quite spread across. On average, they meet either end to end or end to side. What's very interesting is that at the site of contact, no matter where they meet, they end up expressing these transcription factors. So they can meet in all these different fashions and express these transcription factors, so much so that this is both necessary and sufficient. So if we delay bud extension, the expression of these transcription factors is not there. So it is necessary, the contact is necessary. And if we cause the buds to meet precociously in these ablated conditions when the vesicle is smaller, so the buds have lower distance to traverse through, the expression already starts earlier compared to unablated controls. So essentially, these transcription factors, what they are, I told you they're transcription factors. They can affect a wide variety of things. And if we knock them out, it affects pillar morphology. So there were questions about what's inside the pillars. I told you in the beginning that there's this ECM goo in the beginning. And then I told you that this pathway shuts down. Nadia, the graduate student who did this work, showed that this is actually shutting down by activating these transcription factors upon fusion that remodel the extracellular matrix completely. So they remove hyaluronic acid and start to re-express laminin, which was not present earlier in these pillars. Another experiment that I want to show you is this one where I already told you that hydrostatic driven expansion continues during morphogenesis. So now you can imagine that this pillar now is not just experiencing the ECM remodeling inside, but it continues to experience this pressure from the lumen. And you ask like what it's doing. You ask the more question about if the fish is swimming, what it's doing. That would create shear forces in the tube, because at this stage it's already a torus. We don't have an answer to that, but that's a question we want to ask. Here we're asking the question of just, does this hydrostatic pressure impact the pillar morphology? So we can ablate these auric vesicles, release the pressure, and that makes the pillar shorter and wider. So it's a geometric transformation that happens when we release the pressure. We can also do the reverse. We can make these auric vesicles expand using forciclin, which is a cyclic AMP agonist. It activates certain ion transporters, and in this case, the pillars become narrower and thinner. So pressure can change the morphology here significantly, so much so that if we plotted the aspect ratio over different lumen volumes, it scales very nicely. Another thing that I want to highlight here is that the buds are, I told you they're meeting at different fusion zones, but that is also a consequence of the angles at which they're meeting. There is a large amount of variability in the angles at which they're meeting. And the pillars they form also have these wide variety of structures. What we find is that they all get resolved over time. And this, again, we are not using time here, but we're using lumen volume as a metric of time to be able to plot a lot of these pillar straightness angles, and we find that this variability dramatically decreases. Now, the question we're asking is this driven by hydrostatic pressure? Is the hydrostatic pressure responsible for resolving these large variability in pillar structures that we see? We don't have an answer to that, but we have data that supports that we can play around with the lumen volume, and that changes how fast or slow the pillars straighten over time. Okay, I went way over time, but I just want to summarize that this entire morphogenesis is happening. Basically, all the forces are coming from the outside. In the beginning, the buds are extending from this ECM driven pressure. Then this ECM remodels once they form the pillars, and the lumen continues to expand to shape these pillars, or refine the shapes of the pillars later on. But ultimately, I think we all care about about function, at least in this audience, and we are beginning to look at function of these, at least the mutants we have in the lab. We have lots of mutants at this point where the canals are either smaller or the whole aortic vesicle is smaller, or they're completely absent. And in this case, we can very reliably measure behavior as early as in these cases in 5 or 7 days. So we can startle the fish, and a wild type fish would correct itself when we startle it. There are different ways to startle a fish. But if you have mutants where either the canals are absent or smaller, then they exhibit these circling behaviors, which is very reflective of their oversensitivity if the canals are wider or misshapen. So we're beginning to do these experiments in the lab and thinking a lot about what are the constraints and feedbacks in this system. Okay, I'm going to skip this because we all know what that framework is. We all love it, but I just want to thank the amazing team I have and the funding resources. Yeah.